All right, this week we're going to continue on with diversity by getting into the domain Eukarya. And specifically, we're going to talk about the kingdom Protista under the domain Eukarya. But since we're in the domain Eukarya, we are moving away from prokaryotes and are into the eukaryotes now. So if you can think of all the characteristics of a eukaryote, it's going to apply to all these organisms. Okay, so it's important to point out that the kingdom Protista is a very diverse group. And there's really not too many unifying characteristics of all these different organisms that are under this kingdom, except that they're all eukaryotes. So, really, the only classification to put an organism in the kingdom Protista has to be that they're a eukaryote and they're not an animal, fungus, or plant. So, we have our own kingdoms for Animalia, Plantae, and fungi, and that's because those groups are so diverse in themselves. So really the only thing keeping a protist is that it's a eukaryote that is not one of those three things. All right, just some general characteristics that you should know about Protista. They can come in all shapes and sizes. Some of them are unicellular, while others are colonial, while some are multicellular or simple multicellular. And I don't know if you remember from before, but the major difference between colonial and multicellular. And with colonial organisms, even though cells can be bound together and working together to survive, in a colony, each one of those cells can survive on their own. It might not do as well, but it could still survive on its own. Versus a multicellular organism where that cannot happen. If you take one of the cells away from a multicellular organism, it will die. Every single cell needs the other cells to survive. Or just like I said before, all the protists are eukaryotes. So they do have membrane-bound organelles. They have nuclei. They have all the characteristics eukaryotes would have. Now, as far as feeding strategies go, there is a lot of diversity in this kingdom. So a lot of them can be autotrophs, but a lot of them can be heterotrophs, and some of them are even both, can do both autotroph and heterotroph. So if you remember, there's no unifying characteristic to this kingdom, except that they're all eukaryotes and they're not animals, fungi, or plants. And animals, fungi, and plants all evolved from protists. So if we're looking at Protista as a kingdom, this is actually a paraphyletic group. All right, so looking at this tree here, everything under the domain bacteria is found in blue, while everything under the domain archaea is found in red, and then in the brown is where we have our domain eukarya. And in this domain, you could see that animals, plants, and fungi are excluded from the grouping of Protista. However, all the rest of the eukaryotes fall under this kingdom. So when we highlight everything that is a protist, you could see that it is a paraphyletic group. The ancestor to all these organisms, the first eukaryote, was a protist. And then everything that is not a plant, fungi, or animal is also a protist. So we have some of the descendants and the common ancestor here, so paraphyletic. So as we go through this, we're going to look at the major lineages of Protista and the ones that gave rise to the other kingdoms, so fungi, Animalia, and Plantae. And when we're talking about lineage, we're not talking about a specific ranking. So just like we talked about with the phylogenetics lab, we have these taxonomical rankings that we can organize organisms into. However, those are not the only possible groupings we could have. For example, we could find a synapomorphy that can group organisms that would create a group that's between a phylum and a class. That is totally a possibility. So when we're talking about the lineages, we're really talking about different types of groupings, not necessarily the ones that fall in these taxonomical rankings that we're so used to hearing. Okay, so let's quickly look at the 7 major lineages of protists and the ones that gave rise to the other kingdoms, the other 3 kingdoms under Eukarya. All right, so the first lineage we're going to look at is Amoebozoa, and the synapomorphy that allows organisms to fall into this grouping of Amoebozoa is that they have amoeboid-like movement. So we're going to look at examples of what amoeboid-like movement is, but in general, what it is pushing your plasma membrane forward in order to move. The next one we're going to look at is Opisthocanta, and the synapomorphy for the organisms under Opisthocanta is a rear flagellum. So the protists that are underneath this lineage have a rear flagellum, but these protists are the ones that gave rise to animals and fungi as well. And as humans, we still have this synapomorphy, and you might be able to guess where we find this synapomorphy in a human's life cycle. So if you think about sperm, that flagella that we have is a shared characteristic that we have with the rest of the protists under Opisthocanta. So we still have that characteristic. It's just reduced in a very specific part of our life stage. All right, the next lineage we will look at is like Xcauda, and this is characterized by having an excavated feeding groove. So a lot of these are going to be single-celled, and they're going to have this groove inside the cell where they bring in food. So these are mostly going to be heterotrophs. There are autotroph examples, but with this feeding groove is how food is going to enter the cell. The next lineage is Plantae, and under the lineage Plantae is the Protist that gave rise to plants. So plants actually fall underneath this lineage as well. So the kingdom Plantae is just the plants that we're used to seeing, while the lineage Plantae is terrestrial plants like we know, the kingdom Plantae, and all the protists that were related to those original protists that gave rise to plants. So there's a lot of characteristics these organisms have in common. We'll talk about them when we get to it. All right, the next lineage is Risea, and this is characterized by having root-like pseudopods, which could be kind of confusing until you see a picture of it. And really what we're talking about here is very long and slender pseudopods. And we'll talk about what a pseudopod is in a little bit because we also find pseudopods in Amoebozoa. All right, the next lineage is Alveolata, and these are characterized by having alveoli on their cell membranes. And alveoli in Latin means sacs. All right, so you can think of little tiny sacs that are in the cell membrane of these organisms. Now, a lot of times these are unicellular, so one cell. And underneath the plasma membrane, you're going to find all these little vacuoles that are spaced throughout right underneath the plasma membrane. These are the sacs that they're talking about. So this is a unique characteristic to alveolata. And then the final lineage that we're going to cover is Stramonipola, and these are characterized by having hairy flagella. So, like a lot of other organisms, it's going to have a flagella, but you're going to see little fine hairs coming off these flagella. Okay, so it's definitely important for you to understand the major characteristics that define these lineages and kind of what organisms we're going to see in these lineages. Okay, for this video specifically, we are only going to talk about Amoebozoa, Xcauda, Risea, and Alveolata. I've already talked about Opisthocanta. We're not going to look at any examples of that. And then in the next video, in the next class, we're going to cover more of the plant-like protists. So we'll talk about Plantae and Stramonipola next time. So today we're just focusing on these 4 different lineages. And as we go through this, it is important for you to understand the examples that we use. So we're going to see different classifications as examples. So if we look at a genus, you should be able to identify that genus. Also, we're going to look at a lot of the unique structures some of these organisms have, and it's important for you to understand some of these structures because it's what makes these organisms unique and what helps them fall into these categories and how we know that they're related to each other. So pay attention to the examples that we use and the structures that are unique to these organisms. All right, so we're going to start with the lineage Amoebozoa, and if you might remember, the synapomorphy there is amoeboid-like movement. And the first example we're going to look at for Amoebozoa is amoeba. So there is a genus called amoeba, and this is what an amoeba looks like. So amoebas are single-celled organisms that are heterotrophic, meaning they have to go and eat their food somehow. And amoebas are interesting because of how they move. So they use amoeboid-like movement, meaning they push their plasma membrane forward and they kind of inch their way along as they're going through whatever medium they're going through. Now, when they push their plasma membrane forward like this, they create a structure. And we have a name for these structures. We call them pseudopods or pseudopodia. And a pseudopod, it really is just a plasma membrane being stretched out to create almost a foot-like structure. And that's exactly what pseudopodia says in its name. Pseudo meaning kind of, sort of, and podia referring to foot. So this is a foot-like structure. It's also important to note with these organisms, because they have to move in this way, they have to be able to stretch their plasma membrane out forward pretty far. They do not have cell walls. Cell walls are always on the outside of the plasma membrane, and a cell wall would inhibit their ability to move. All right, so now we're going to look at a video of an amoeba to see if you can identify some of these structures that we just talked about with the genus amoeba. Okay, what we're looking at here is an amoeba, this single-celled over here, about to eat these 2 paramecium that are off to the side. And it did this by expanding that pseudopod around them. So pseudopods aren't just used for locomotion, they're also used to consume things. So let's watch that again. As it stretches this pseudopod, it's expanding the plasma membrane forward and then engulfing these two paramecium that are here. So you can see the pseudopod being pushed forward, the plasma membrane is being pushed forward, and then the paramecium are engulfed. And then what happens once the paramecium are inside the cell? The amoeba is going to release digestive enzymes to break down these paramecium. And you will know when that happens because the paramecium do not like it. They immediately go into this alarm state because they know they're being digested. Okay, so let's look at this again and actually see how the amoeba is making the pseudopod. And again, this is one giant cell. And how amoebas make these pseudopods is by pushing their cytoplasm, squirting it into the plasma membrane to create this kind of bubble in the plasma membrane, the pseudopod. And we have a term for this. We call this cytoplasmic streaming, meaning streaming the cytoplasm into the plasma membrane to kind of push out this pseudopod. And you could see the cytoplasm being pushed into these pseudopods right here that are being formed. All right, so that is how they're doing this through cytoplasmic streaming. Okay, so the next organism we're going to look at that also falls underneath the lineage Amoebozoa is Physarum. So the genus is Physarum, and Physarum is a genus that falls under a specific grouping of plasmodial slime molds. Now, for a long time, people thought these were related to fungi in some way, and that's really where the mold name is coming from. They thought they were fungi, but they're actually not. They're more closely related to amoebas than they are to fungi. And these organisms have some very interesting characteristics. One of them, and one of the most interesting characteristics, is that you could see these organisms with a naked eye. But what you're looking at is actually a single cell. These just have a giant cells that you could see macroscopically. That is very rare. So what you're looking at is one a giant cell. There's only one membrane that surrounds this entire organism. So there's no separation anywhere in the cell with a membrane. And another interesting characteristic is that they are multinucleated, meaning they have multiple nuclei spread throughout this giant cell, which kind of makes sense because if you're going to cover this much area, you're going to need different starting points to produce different proteins in order for this organism to survive. Now, these organisms do make something called sporangia, which are going to be these little structures that stick upwards to release spores. And just like amoeba, they have cytoplasmic streaming to push their plasma membrane forward. So they also have this amoeboid like movement, using cytoplasmic streaming to push their membrane forward to kind of creep outwards from a central location, looking for food. So these are not autotrophs, they are heterotrophs. They are out there looking for food to digest. Okay, so here is a video of that cytoplasmic stream. You can see them pulsating their cytoplasm forward at the edge of this plasma membrane that's out here, pushing it outwards. So you can see this organism radiating outwards, looking for food. And once it grows outwards, it still stays connected with every other piece of this cell. So it is making sure it's passing these nutrients throughout the cell, whatever nutrients it finds. But you can see this pulsation, this cytoplasmic streaming that is going on, pushing the plasma membrane outwards. Okay, so another unique characteristic of Physarum is that it creates a sclerotium, which is kind of this dormant stage. So these things are not moving around like we saw with the amoebas. They're kind of stationary and just kind of growing outwards. And eventually they might run out of nutrients. And when that happens, they can go into the sclerotium stage, this hardened stage, and lie dormant and wait for nutrients to return to whatever area they're growing. So let's take a look at the sclerotium being formed. So here's a Physarum that is growing on an auger. So an auger is just a gel with a bunch of nutrients in it, but you could see with this auger, as this Physarum absorbs all the nutrients, it's going to start turning into this hardened mass. So as the nutrients are disappearing, this single cell, this very large cell, is hardening to go into this dormant stage. And it will stay in this crusty stage until nutrients return. Now, if you want to learn more about these organisms, I put this video in here that you could follow. It's pretty long, it's a TED Talk, but it's very interesting because these organisms are used for a lot of different studies. These organisms actually show some level of intelligence, even though they're a single cell. Or if they've done studies where these organisms can actually keep track of time intervals, and these organisms are always finding the fastest route between two areas in order to get to the nutrients quicker. And so a lot of scientists will use this as their study organism because they show all these unique characteristics, even though they're a single cell, they're one cell. Okay, so the next lineage we're going to look at is Xcauda. And Xcauda, remember the synapomorphy of this lineage is an excavated feeding groove. So again, we're going to look at single-celled organisms here, except for they're going to have some sort of groove on the side of their body somewhere in order to intake food. Now we're going to look at two major examples for Xcauda. The first one is the class Parabasilia, also known as the Parabasilids. And this is a unique class of excavates that have evolved to live a symbiotic relationship in the gut of different animals. So you will find these organisms in the guts of animals. Most notably is with the termite. So we all know that termites eat wood, but it's actually not the termite that is digesting the wood. These things are living in the termite's gut, digesting the wood for the termite. So they have this animal that is housing them, giving them a nice, safe environment to live in, and in exchange, they're breaking down wood for that termite. And even more interesting is that these organisms aren't really the ones breaking down the wood, but it's actually the bacteria that are found inside the cells of these organisms that are breaking down the wood. So in this organism, they have bacteria that contains an enzyme called cellulase that is able to break down the cellulose of wood, and the nutrients that are released are then given to the parabasilid, which can then give those nutrients to the termite. So here are three examples of a parabasilid. So if we were actually meeting in class, we would do this experiment by popping open a termite's guts and actually looking at these organisms. All right, but each of them move very differently. So Trichomonas kind of has this whole pulsating wave that goes on when they're moving, versus Strablomastics, which is more of a worm and it kind of corkscrews around the termite, versus Trichonympha, which has these long like hairs coming off the cell to help it move around. So let's see if we can identify these. So what we're looking at here is a microscope image of these different parabasilids that are found in a termite gut. So the termite has been spread across a slide, and now we're looking at these organisms in real time. And you could see all three of those genuses here. So as we hit play, you could see the Trichonympha with its very long hair-like structures. You could see the Trichomonas with its wave undulation around the cell. And you can even see the Strablomastics, all these little corkscrew long organisms that are swimming around everywhere. So there's actually a lot of diversity going on in the termite gut. But what they're all doing is helping the termite break down the wood by using cellulase, that enzyme that breaks down cellulose. So the next example we're going to look at is the euglenoids or euglenids. You could use either term. And these also fall under the lineage of Xcauda because they have an excavated feeding groove. And specifically, we're going to look at the genus Euglena. So euglenoids fall under the phylum Euglenophyta. All right, so they all have similar characteristics that are underneath this phylum. But let's look at some of the characteristics Euglena has, this genus Euglena. Okay, one, Euglena has something called a pellicle. And what a pellicle is a cell wall. So this is, again, a single-celled organism. But they have a cell wall that goes all around it that is very flexible. So that's kind of unique compared to other cell walls we see in different organisms. So most of the time, cell walls are pretty rigid. But here with pellicles, they're actually pretty flexible, giving some protection to the organism, but also allowing it to move and bounce around. Okay, knowing it's part of Xcauda, we know it has a feeding groove, and you could see this feeding groove, this reservoir right here. So it's going to take in food this way. So knowing that, is this a heterotroph or an autotroph? Well, it's a heterotroph. It does eat food. It can eat food, but it's also an autotroph. This is one of those unique situations where we have both. And you could see all these different organelles that are floating around in here that are green. These are actually chloroplasts. So they're going to use chloroplasts to photosynthesize and then use their feeding groove to eat things that are swimming around. All right, these also move with using flagella. So a lot of times coming out of this feeding groove of this reservoir, you're going to see flagella sticking out. And they have two flagella. They have one long one and one short one that usually does not come out of the reservoir. And they also have this structure here called a stigma or an eye spot. And what this is a very rudimentary eye. It can detect light. Now, why would that be important for these organisms? Well, they do photosynthesize. And the preferred method of feeding is through photosynthesis. It's the more efficient way to eat. So they want to photosynthesize first. So they'll use this stigma, this eye spot, to find where the light is and swim towards the light source. But then sometimes the light source isn't there, like at night. When that happens, they will stop trying to swim to the surface and photosynthesize and actually switch their diet to a more heterotrophic diet, looking for food. Okay, so let's look at this video of these Euglena to see if we can identify some of those structures we just talked about. All right, so we have this flagella sticking out. We have another one in here that is more reserved, and you can kind of see the feeding groove right here where the flagella is coming out of. But you can definitely see the stigma, that eye spot to help it see light, detect light, and you can see all these chloroplasts that are floating around it. And even look at some of these Euglena, they are really changing their shape, and they're able to do this because of that pellicle, that really flexible cell wall they have. So even though they have that protection of a cell wall, it's extremely flexible, allowing them to move around and become more amorphic. Okay, moving on to the next lineage, we're going to talk about Risea. And if you remember, Risea has that synapomorphy of root-like pseudopods. So we saw pseudopods in amoebozoa. It's really when you push your plasma membrane out. Well, these organisms do the same thing, except it looks a little different. Instead of having this foot that they stick out of their plasma membrane, they actually have very long threads that they make from their plasma membrane. And I'll show you a video of that in a second. The example we're going to use is Foramnifera, which is a subphylum, and short term is just forams, so you can refer to them as the forams. And with these organisms, they are heterotrophic. So these are single-celled organisms that are heterotrophic, and they actually make a shell around their cell. So they have a calciferous shell, meaning it's made out of calcium. So these very tiny microscopic organisms that make really tiny shells around their single cell. Now, these have been around for a long time, and you might have heard of the White Cliffs of Dover in England. This is a deposit of these organisms. This is millions of years of buildup of these organisms growing, blooming, and dying, and leaving their calcified shell behind. So that's what we're seeing here in the White Cliffs of Dover. This is millions of years of buildup of these shells. So let's look at a video of an alive foram to really see this root-like pseudopod that they create. Okay, so here is one of those forams. So this is a single cell that's living in a shell that it's created, and you could see all these projections coming off of it, sticking away from this organism. So all these little projections are actually pseudopods. All right, these pseudopods look very different than the amoeboid pseudopods. So this is the plasma membrane sticking very far out in a thin line, like a root. So that's where the term's coming from, a root-like pseudopod. And it's going to use these pseudopods to feel around for food because these are heterotrophic organisms. They are not autotrophic. So they're out there actively looking for food using these long projections. Okay, so this was our only example for Risea. So next we're going to move on to Alveolata. And with the lineage Alveolata, we have those alveoli, those little sacs that are underneath the plasma membrane of these organisms. So again, these are going to be a lot of single-celled organisms. And one of the examples we're going to look at is the ciliates. So ciliata is a class, and one of the examples of a ciliate we're going to look at is a paramecium. And because paramecium falls under this grouping of ciliates, is from the structures that they have all around their cell. These little tiny hair-like structures. This is referred to as cilia. So we have these hair-like structures all the way around the cell that it uses for locomotion and for creating water current. Now, this is very different than the flagella that we saw in other organisms. Flagella are usually only a few and very long and whip-like, while cilia has many and are more hair-like and cover the entire cell for the most part. Now, with paramecium specifically, they have a lot of unique characteristics that you won't really find in other organisms. And there are a lot of characteristics that paramecium shares with Euglena, even though they are completely different lineages. So looking at this tree, we have our lineage Alveolata. Here's our ciliates, where we're going to find paramecium. And notice where we're going to find the Euglena. All right, Euglena is way over here in this lineage. All right, but even though they're so distant, they still have a lot of similar characteristics, a lot of them mostly from convergent evolution. Okay, so one of the characteristics that they have that they share with Euglena is a pellicle. They also have a pellicle. And if you remember, a pellicle is a cell wall that is flexible. So that gives these organisms a lot of flexibility by having the cell wall. Another characteristic they share with Euglena is that they have oral grooves. So these are heterotrophic organisms. And they have an oral groove, this time located on the side of the cell, that they can use to intake food. So they're going to use this cilia to kind of beat a current and bring food into this oral groove. Now, with Paramecium, they are strictly heterotrophs. They do not have any chloroplasts or anything like that, so they're strictly heterotrophs, while Euglena were both autotroph and heterotroph. All right, something else that's pretty unique to Paramecium is that they have two nuclei. They have a macronucleus, which is usually found in the center of the cell, and then they have a micronucleus, which is found off to the side. And these are going to house different types of genes. So the macronucleus is going to house the genes for most of the normal metabolic activity this organism needs to do in order to survive, while the micronucleus is actually going to house the genes for sexual reproduction. And the type of reproduction Paramecium undergoes is something called conjugation. We might talk more about conjugation later on in this course, but for right now, just know this is not a very common form of sexual reproduction. However, Paramecium is able to do it, and the genes for conjugation are coded inside this micronucleus. Another important organelle that these organisms have is a contractile vacuole. So you might see these contractile vacuoles off to the side, and this is going to help with osmoregulation. These are aquatic organisms, and to prevent bursting from too much osmosis, they have these organelles that can help regulate that, regulate the osmolarity that are inside the cell. So again, let's take a look at a video, see if we can identify some of these structures. Okay, here we are looking at Paramecium. So again, Paramecium are heterotrophic. They do not have chloroplasts, even though some of you might be thinking that these look like chloroplasts. These are actually food vacuoles. So they do eat other autotrophic organisms, and when they digest it, they store it in these vacuoles. So you're really just seeing the digestive remains of whatever they ate. Something else you can notice here is their cilia. Look how they bead their cilia to create these currents to move around and to actually force food into their feeding groove. Two things you probably won't see are the macro and micro nuclei. And that's because nuclei typically are transparent, and you can't see it without adding some sort of stain to the slide. And if we add a stain, then we'd kill this organism, and then we wouldn't really see a lot of the other structures here. And you can even see some of these tiny alveoli that are underneath the plasma membrane. You see all these little tiny sacs that are all around this organism. That is the synapomorphy for this lineage. And here we're looking at those osmoregulating organelles, those contractile vacuoles right here, opening and closing, helping to keep some sort of homeostasis with the osmolarity of the cell. Okay, so the last organism we're going to talk about in this video is also underneath the lineage Alveolata. So they do have those little tiny sacs that are underneath the plasma membrane. And here we're going to look at dinoflagellates. Now, dinoflagellates are a pretty diverse group, and they all fall underneath the phylum Dinoflagellata. Some dinoflagellates are autotrophic, while some dinoflagellates are heterotrophic. You really won't find one that's both. So they either have chloroplasts or they don't, and they go and find their food. So the examples that we're going to look at here are all autotrophic, meaning they are able to produce their own food using chloroplasts. Again, these are single-celled organisms, and here they make a cell wall that is known as a theca. They have this armoring that is around their cell, while some of them don't make any cell wall and are referred to as naked. So here's two examples of that. Up here we have Carenia brevis, which is a naked form of dinoflagellate. So you have this one cell here that has no armoring around it, versus down here with the Serratium, which is another type of dinoflagellate that does have an armoring or a theca around it. So the reason we bring up Carenia brevis is because it's a very relevant organism in Florida. Carenia brevis is one of the major types of organisms that produces red tide. Now, you might have heard of red tide before. This is a very common problem in Florida, mostly on the west coast of Florida in the Gulf of Mexico. But sometimes you can find these blooms in the Atlantic as well. And red tide is really bad for two reasons. One, Carenia brevis makes toxins. They make a suite of toxins called brevetoxins that are able to aerosolize. So these toxins are getting into the water here, killing off fish, and they can also get into the air and cause problems with people who are around here that are breathing in those toxins. So these red tides are responsible for deaths of a lot of different organisms. We would see mass fish kills near red tide events. We also see marine mammal beachings after red tide event. So when these blooms occur, we see a lot of different organisms perish, and we'll see them washed up on shore eventually. Okay, so the last example we're going to look at for dinoflagellates is zooxanthellae, which is a dinoflagellate that you may be familiar with. And zooxanthellae is a dinoflagellate that has a symbiotic relationship with coral. So here is a close-up of a coral polyp right here. And so you might be familiar with coral since it produces that calcium carbonate skeleton that makes up reefs, coral reefs that we have so many of here in Florida. But something you might not know, when you think about a coral reef, usually you think about how colorful the reefs are. Well, those colors are not coming from the coral. Those colors are actually coming from the zooxanthellae that are living inside the coral. So all these little tiny dinoflagellates are actually living in the tissue of the coral, and that's what's giving it its color. All right, so what's the relationship here? Well, dinoflagellates, the zooxanthellae, are photosynthetic. So they're going to photosynthesize and give some of those sugars to the coral. Well, coral are heterotrophic, and they're also eating things that are swimming past the coral polyps, and they're going to release nitrogen and other types of waste that the zooxanthellae can use. So this is a very tight and mutualistic relationship. And when you hear of coral bleaching, what is happening there is that the coral are getting stressed and releasing all their zooxanthellae. So the reason these coral look bleached is because they lost the zooxanthellae, which is what's giving them their coloration. So all you're seeing is this transparent polyp and the calcium carbonate skeleton underneath and that's why they are white. And this is an obligate relationship, meaning once these zooxanthellae are gone, the coral will not survive long without them. They need these algae to survive. And if they go too long without these algae, they will die. Okay, so that's everything we're covering in this video. Just a couple things to keep in mind as you're going through the material. One, make sure you can identify any genus or other examples that we use for each one of these lineages. Also, make sure you can identify any of the structures and have a general understanding of what they do for any of these organisms that we looked at. Finally, definitely understand what characteristics these organisms have that make them unique, and even some of the similar characteristics they might have with each other. Next, definitely understand the lineages that they're coming from, and definitely understand the synapomorphies that make up those lineages. So we talked about that in the beginning, but it's very important to understand how these organisms are classified and how they're related to each other. And finally, just keep track of your taxonomy. So we've talked about different phylums, classes, genus, all this other stuff as we went through this video. So just make sure you understand what taxonomical ranking we are talking about with each one of these terms. All right, so hopefully this helps clarify some of the diversity that we see with these protists. We're going to come back to protists and finish up the different lineages that we did not cover in this video.